everyone. So I thought we'd start off today's lesson by going over the homework. That way, I know some of you already asked me questions about the homework. The directions for number 1 are a little bit confusing, so I'm going to explain them verbally now. But I did write a post on Google Classroom to clarify. Also, somebody asked a question in the extra help document, and I clarified it again in there. So please make sure, even if you don't have a question, that you take a look at that extra help question document, because you may not realize you have a question until you see that somebody else asked it, or you may have a question and somebody else may already asked it and then gotten an answer from me. So just, get in the habit of looking at that extra help document. I'm also going to, instead of commenting on every single one of the questions from Edpuzzle, I figured that it would be helpful to just have a document with all the questions from Edpuzzle, specifically the ones that people seem to struggle on, and then have the corresponding correct answer to just clear up some of the common misconceptions I've been seeing. Again, instead of commenting on every single one of your Edpuzzle posts individually. So we'll try that. And again, feedback is appreciated, so let me know if you think that's a good idea, okay? So let's just go over this homework. On Friday, we talked about how to determine the 5' end of DNA strands versus the 3' end of DNA strands. So if we look at question number 1, this is a representation of DNA, right? If we look at it, we have DNA. Why? How do we know it's DNA? Well, it's double-stranded, besides the fact that it says double-stranded DNA, but DNA is a double helix. Also, it tells you the sugar type, which is deoxyribose. And if we look at the nitrogenous bases, we see thymine, another indicator that it's DNA. So we can, split this DNA in half. We're going to look at each strand individually to determine the 3' end and 5' end. So we're going to label this strand number 1, strand number 2. So it tells you to circle and label the 5' end carbons, the end 5' carbons, and 3' carbons, right? So in terms of circling, right, how are you going to determine where the 5' end and 3' end of DNA are? Looking at the carbons. So here we have our first full nucleotide. What's a nucleotide? A monomer of nucleic acids, a monomer of DNA and RNA. It's made up of what? It's made up of let's fix this. Made up of a nitrogenous base, made up of a 5 carbon sugar, made up of a phosphate group. So here we have our 5 carbon sugar. Okay? Now let's, even though it's already labeled, count the carbons. So oxygen's at the top. Then, like it tells you in the directions, that reminder, where 2 or more lines meet, that means that there's a carbon there. So even though this is numbered 1, if we look at this picture down here, right, there's a carbon here, a carbon here, a carbon here, a carbon here. So when we're looking at this structure where 2 lines meet, that symbolizes that a carbon is there. So let's take a look at this first carbon. Start at the oxygen, go clockwise. 1', 2', 3', 4', 5'. 5' always connected to a phosphate group. So in order to determine what end of DNA is the 5' end or the 3' end, you look at the carbon that is hanging off. In this case, I would circle this 5' carbon and label this the 5' end of the DNA strand. DNA is what? It is anti-parallel. So that means if I already labeled this end of DNA 5', I'm going to assume that the other beginning strand of DNA is 3'. But how can we tell? Well, this side of the DNA, the 5-carbon sugar, the deoxyribose, is now upside down. So instead of starting with oxygen facing off of the strand, now it's down here. But we do the same thing where we start at oxygen and we're going clockwise. That means that this is 1', 2', 3', 4', and 5'. What carbon is hanging off of the DNA? The 3'. 3' carbon always attached to an OH group. This is the 3' side of DNA. Look at, they're anti-parallel, right? This side starts with 5'. The number 1 strand starts with 5'. The number 2 strand starts with 3'. And again, anti-parallel, so we can already assume what's going to happen on the bottom. Look at this final carbon down here, figuring out what side of the DNA it is. 1', 2', 3', 4', 5', which is right here. So 3' hanging off the end. This is the 3' side of DNA. And we look over here. Same thing. 1', 2', 3', 4', 5'. Which one's quote unquote hanging off of the DNA strand? That would be the 5'. Now, the second part hopefully wasn't too hard for you. It gives you this strand of DNA. This would be our original strand, so we're going to call this our parent strand. And we want to draw in the complementary DNA bases that would form. It tells you to draw it above. I'm just going to put it up below because I have more space. But this is our original parent strand DNA. And our daughter strand, I'll do it in a different color. Daughter strand is the copy that we're making. How is DNA copied? Well, it's going to be copied in the 5' to 3' direction. So this parent strand runs from 3' to 5'. And we're going to be creating the daughter strand from 5' to 3'. They run anti-parallel. Now we're talking about DNA here. So our bases are adenine, bonded, hydrogen bond with thymine, and cytosine bonded with guanine. So you can remember apples in the tree, cars in the garage. We're talking about DNA, so thymine, not uracil. So if we write in the daughter strand, the complementary daughter strand, adenine bonds with thymine, guanine, cytosine, cytosine, guanine, guanine, cytosine, cytosine, guanine, cytosine, guanine, thymine, adenine, thymine, adenine, adenine, thymine. Copying from 5' to 3'. So that would be the anti-parallel strand of the DNA.